share my screen with all of you. All right, so welcome to this lecture, everyone. Can all of you see the larger screen? Can all of you see that well? Okay, awesome. So we're going to be talking about the auditory system. Special thanks to my friend, Dr. Brad Walters, for providing this image of a mouse cochlea. I just love it. All the rainbow colors, very nice. And so first off, I want to pose this quote for all of you. When you lose your vision, you lose contact with things. When you lose your hearing, you lose contact with people. So does anybody have an idea of who said that? All right, how about now? Anybody know who this is? All right, this is Helen Keller. All right, so can any of you imagine what it's like to live without your vision or without your sense of hearing? So you're essentially living in a world of darkness, you know, kind of adrift in this world of silence. Personally, I know I cannot imagine that, but what I'd like all of you to do for right now is just try to at least imagine life without your sense of hearing. So you would not be able to listen to music. You would not be able to understand the tone or the mood of the people that are speaking to you. You'd no longer be able to hear the voices of your loved ones. And you would never be able to hear another lecture again. The horror! So I really feel like Helen Keller's words, though, ring true. And there's really this profound sense of experiential truth that really only she is privy to, you know, because we walk around, we take our senses of sight and hearing for granted. So it's really a humbling thing to consider. So for this lecture, we're going to visit just learning objectives really quickly. And then the main body of the lecture will talk about transduction of sound, the auditory system pathways, and hearing loss. We probably won't have time to really get to the vestibular system because that is an entirely different system that doesn't really, it's somewhat related to the auditory system, but the vestibular system is our system of balance. So I do have some slides at the end, but we probably won't get that far. Now, through the lecture and through the slides that are already attached in Blackboard, you'll see some content that says FYI for your information. That means you're not responsible for really learning that information, and I don't expect you to learn it. It's more so for your interest. And what I'll try to do is also make note of any detailed content throughout the lecture that I don't really expect you to memorize or try to learn in full detail. The auditory system is arguably the most complex system in the body, especially in the neural network of systems, in my opinion. And so paradoxically, it's very easy to miss the complexity and the intricacies of the system, while at the same time, it's very difficult to try to capture really that complexity and appreciate it in a way that's easy to understand. In this lecture, there will be a few case vignette questions that we can try to use as practice as we go through the material. I know y'all will not be clinicians. However, if you go into academia, you might be teaching future health professionals one day. And if you're going to be teaching future health professionals, you will probably have to teach courses and write test questions. So you might as well get some experience and exposure to it. So for today, we're going to talk a little bit about the range of human hearing very briefly. And then we're going to talk about how sound waves travel through the ear and how they're ultimately used to transduce sound, and all of those outer, middle, and inner ear structures that are involved. From macroscopic to the microscopic level. We'll also talk about all the other anatomical structures and the auditory system pathways, all the way from the hair cells to the primary auditory cortex. And we'll apply the use of what's called the ABR, or the auditory brainstem response, to localize lesions or any type of damage along the auditory system pathways. We won't talk about the Wren and Weber tests. Those are more so used by clinicians, but I did include those slides in Blackboard so that you can look at those. These are tests that are mainly used by people that live in rural areas, so clinicians that might not have access to sophisticated equipment like the devices that are used for registering or measuring the auditory brainstem response. All right, so let's talk about sound transduction. Before we do that, though, let's visit the range of human hearing. So sound is a vibration wave, and you've probably visited this idea in physics or physical science. And whether or not we hear that vibration depends on two things. That is intensity, or the measure of the sound power per unit of area. So this is the peak to trough amplitude. So if you look in the image here, the diagram, you'll see the letter A representing this distance between the crest or peak and the trough. And so it's that amplitude of force at the tympanic membrane or the eardrum. And we'll talk more about that later. So intensity is not the same as loudness, but it is related, and they are both measured in decibels. The second characteristic is the frequency. So this is the measure of the number of cycles per second in which a sound wave travels. So another way to think about this is the number of waves that passed a fixed point in 1 second of time. So that's measured in cycles per second, also known as hertz. So if you can imagine this line, vertical line, going right down the middle of our diagram here, you can imagine that being some arbitrary point. So the number of cycles would be the number of waves that pass that point in 1 second in time. All right, so we can only hear vibrations within a certain intensity range and a certain frequency range. And we can see some examples of audible sounds that we come in contact with maybe in our daily lives or from time to time occasionally. So if you look at the line graph here, you'll notice that all these audible sounds are above this curve because they certainly fall within that intensity range that is necessary for us to be able to detect those sounds. So the lowest threshold of hearing is around 2000 hertz in frequency. So as I mentioned earlier, anything that is lower than an intensity level of 15, we won't really be able to hear that sound. So if you notice here, all of our intensity levels are higher below that frequency level threshold and above that frequency level threshold. And so that's necessary for us to be able to detect those sounds. Now, a little bit more useful information. Our normal range of hearing is anywhere from 50 to 16,000 hertz. And most speech occurs within a window of about 100 to 8,000 hertz. And that is about 50 to 60 decibels of intensity. And we are routinely exposed to a range of about 85 to 100 decibels of sound intensity, and anything over 120, say, for instance, like a rock concert or the sound of airplanes taking off, that exposure over prolonged periods of time can actually lead to hearing damage. Now, that we've talked about the range of human hearing, we'll talk a little bit about sound transduction. And I encourage you to look in those files in Blackboard. You can actually click on the link to watch this video. I won't play it for you, but it would be a nice video to watch after the lecture to kind of put together all the pieces of the process if some of these aspects don't make sense to you. So to begin, we've got to start with the anatomy of the ear. So we have the outer or external, middle, and inner ear. The key to all of these structures are in the note slide. So the note section of this particular slide, so that you can refer back to for your reference. But let's focus on the outer ear. So the pinna, number 1, or auricle, is this cartilaginous structure that sound waves first come in contact with. They then move through the external auditory meatus more medially. That's represented by number 2. And then from there, they reach the tympanic membrane or eardrum, which is represented by number 3. So all of that is a part of the external ear. Then sound waves will move into the middle ear cavity. That's denoted by number 4. And you'll find some very important players inside the middle ear cavity, and those are your middle ear bones, your middle ear ossicles. 5, 6, and 7. Those are the malleus or hammer, the incus or anvil, and the stapes or the stirrups, because they kind of look like stirrups, like on a horse. So the foot plate of our stapes bone actually comes in contact with our inner ear structures via the oval window. So we cannot see the oval window in this diagram because the foot plate of the stapes bone is covering it. But that opening actually opens into this bulbous structure denoted by number 8 called the vestibule. Hence the vestibular system. You know, it all makes sense. So the vestibule has connections with both the cochlea, this snail-shaped structure labeled by number 11. Cochlea is the Latin word for snail. And these loop-like structures denoted by number 9, the semicircular canals. So we won't talk about the semicircular canals or the vestibule in great detail, hence their relation to the vestibular system. We will focus more so on the cochlea and its relation to hearing. The remaining structures that we can see in this diagram, number 10, this is representing our vestibular and cochlear divisions of our vestibulocochlear nerve. That's one of your cranial nerves, the 8th cranial nerve. Again, we'll focus more so on the cochlear division. And then number 12, this is your eustachian tube, also known as the pharyngotympanic tube. So this is important for helping with drainage of the middle ear cavity. Now, being realistic, you will probably never see this anatomy in this vantage point. And you probably won't really be ever charged with looking into someone's ear from this vantage point like physicians do when they take an otoscope and they look into someone's ear. But maybe just one of you will end up doing some type of research out there in the world on the auditory system, and some of this knowledge will come in handy. So now let's take our understanding of this anatomy and talk about the movement of sound waves. So for the sake of simplicity, I've removed those vestibular structures. And as you can see here, the sound wave moves through the external auditory or acoustic meatus, hits the tympanic membrane. As the tympanic membrane is depressed, sound waves become mechanical energy as that depression initiates this chain reaction through our middle ear bones, these middle ear ossicles. And then those waves are converted into fluid waves when they travel into the cochlea, okay, which is in the inner ear. And we're going to talk about how that happens a little later. So, got sound traveling through the external ear where amplification of sound is initiated. Further amplification of sound occurs in the middle ear through the help of these middle ear bones. And impedance matching, which we're going to talk about later, also occurs there, so that we can transfer these sound waves into fluid waves into the inner ear, where ultimately our sound transduction is going to occur. So let's go through each of these one by one. So if you've ever looked at your ear, it's got all these weird structures in it. You know, they have all these weird names. And in fact, they all play some role in helping sound get into that small space. So an overly simplistic way of thinking about this is thinking about your ear as though it were a funnel. And another way to think about it is it's like an antenna system. It's an antenna system that helps amplify the sound that we want to be transmitted into our heads through our external auditory meatus. And some of those are actually resonators of sound, those being the concha, the walls of the external auditory meatus, And so they prolong sound vibrations that will ultimately allow us to hear sound better. Now, when we get to the middle ear, I've already talked about the middle ear bones, but you can see in this diagram here on the left, you can see the movements of these structures and the axis of rotation along the joints of those bones where they articulate with one another. So you can see here as the tympanic membrane is depressed and it moves more medially, it's going to cause the manubrium or the handle of the malleus to move more medially. The axis of rotation then is counterclockwise, where the malleus articulates with the incus. And then in turn, the incus will also move medially, pushing the stapes into the oval window. There are two important muscles that are attached to two of our middle ear bones, and we'll visit those in this next diagram. So imagine that you are walking into the middle ear cavity. So like I'm opening up the tympanic membrane and I'm stepping into the middle ear cavity and I'm turning around and I'm now facing the tympanic membrane. So now you are viewing essentially the backside of the eardrum, the tympanic membrane. That's what we're looking at in this colored image. All right, does that make sense to everyone? So instead of looking at the back side of the wall of my middle ear cavity, I've now turned around and now I'm looking at the other side of the tympanic membrane that I can't see if I'm just looking through the ear with an otoscope. Now, what I like about this image is now you can see how the tensor tympani muscle attaches to the manubrium of the malleus, and the stapedius muscle attaches to the stapes bone. So these tendinous connections to these muscles and the contraction of these muscles are what help dampen sound. So these essentially help protect your ear from sounds that are way too intense or way too loud. So without them, we don't have that ability to do that. The other thing I wanted to point out in this image is there's this little nerve. There's a little nerve that kind of courses along the medial superior aspect of the tympanic membrane. Does anybody have an idea of what that nerve is? All right, so if you remember in geometry, you talked about this thing called a chord. So a chord is any line that intersects a circle, you know, crosses a circle at 2 points, but it's not the diameter. Okay, so this is called the chorda tympani nerve because it's the chord of the tympanic membrane intersecting the tympanic membrane, this round structure crossing 2 points. Now, this structure carries taste sensation from the anterior 2/3 of your tongue. Okay, so why is this important? Well, you've already talked about taste. You've already had a lecture on taste. And so any type of surgical procedure that involves, you know, maybe inserting tubes, drainage tubes in the ear that, you know, children oftentimes get, it's important that you do it low so that you don't damage this nerve. Because if you do, then you can affect someone's sense of taste. All right, now let's talk a little bit about impedance matching. So if you notice, the tympanic membrane is rather large, oval window is rather small. So this is important for transforming small force into a larger force. So when sound waves are transmitted from the eardrum to the oval window, the middle ear essentially functions as an acoustic transformer that helps amplify those sound waves as they move into the inner ear. This little equation here kind of simplifies that idea. Large area times small force equals small area times large force. And in fact, it's estimated that there is a 10-fold increase in force per area through this process. So this is going to be important, as I said earlier, for pushing into the thicker fluids thicker than air, inside our inner ear. That fluid is called perilymph, and we'll talk a little bit about the concentrations of the ions in perilymph in later slides. So another way to think about this is imagine someone with a stiletto heel stepping on your foot. Hurts really bad, doesn't it? Doesn't hurt quite as bad as someone with a flat heel, a flat wide heel stepping on your foot. So this is the same concept as to why that smaller area ultimately translates into a more intense force. So we finally made it to the inner ear where all the magic really happens. So we're looking into the cranial vault. So imagine I'm cutting off my skull cap, removing it, and now I'm looking into my skull. Anterior is front, posterior is back. And now I want to focus on this blue boxed area. So this is called the petrous part of the temporal bone. And it's outlined in this dashed green line. It is the hardest bone in your body. Aptly named because petrous comes from the Greek word for rock, hard as a rock. Can you guess why? Why would this need to be the hardest bone in your body? Or why might it be?
Maybe the eardrum is really sensitive and needs to be protected.
Yeah, exactly. So you got really delicate structures in there and they need protecting. Yeah, perfect. Now, the cochlea sits in this area and it's kind of like a spiral staircase. So imagine you're at the top of the spiral staircase where it's wider. You're moving your way down the spiral and it gets narrower toward the bottom, toward the apex. So the base of our cochlea is actually up top. Apex is down below. A little counterintuitive. And here, imagine we're taking a sagittal section through the cochlea. So we're making a cut through the cochlea and we're looking at the inside of these inner coils. Okay, so you can see some of the internal anatomy of this. Don't focus on that right now. We just want to appreciate the orientation in space. So the base is superior or up, apex is inferior down, and then the apex is also pointed slightly more anteriorly. The base is tilted slightly more posteriorly. So keep this orientation in mind as we continue. Now, the inner ear structures essentially consist of a tube within a tube. You've got a bony labyrinth or a bony tube, and these bony structures are all connected, the cochlea with the vestibular structures. And then we have our membranous tube or our soft tissue tube inside. So think of the light blue space here surrounding these membranous structures that are in the pinkish purple. That is the perilymph. That is the fluid between this bony labyrinth, this bony tube, and the membranous or soft tissue tube. Inside this membrane tube, we have special organs, special sensory organs known as the end organs. And those organs are also bathed in a fluid known as endolymph. So endo is more inside, internal. Perilymph is around the outside, inside the bony labyrinth. So we're going to focus on the organ of Corti. That is the end organ inside the cochlea. So what does the cochlea look like under the microscope? Here it is in all of its glory. Again, we have a sagittal section through the cochlea, but we've turned it upside down. Apex is on top, the base is at the bottom, and this is typically how it's viewed under a microscope. So let's hone in on one of these turns of the cochlea and look at that internal anatomy that we haven't really talked about yet. You got three spaces. The scala vestibuli, which is where the cochlea is continuous with the vestibule. You have the scala tympani, which is where the cochlea opens into the middle ear cavity via the round window. Okay, so that's another window that it opens into, and that allows for the equalization of pressure from the sound waves. That's why you have two windows. And then in the middle, we have the scala media. The scala vestibuli and scala tympani are enclosed by our bony labyrinth, our bony tube. So we find perilymph inside of these spaces. This is extracellular-like fluid, so it's very low in potassium. The scala media, in turn, is filled with our endolymph, and it's going to be bathing our end organ, our organ of Corti. This is intracellular-like fluid, fluid like the inside of a cell because it's high in potassium. And this is going to be important for sound transduction. So let's take a closer look at these structures. The structures in red are the more important structures that you want to know. The inner hair cells and outer hair cells can be seen here by their abbreviations. You'll notice that the inner hair cells are closer to the midline of the cochlea, the bony medialis, which is the spiral axis of the cochlear structure, and the outer hair cells are closer to the periphery. There are more of those. They rest on a membrane known as the basilar membrane, and that's going to be one of our moving structures that encodes sound frequencies. We're going to talk about that in just a little bit. The tectorial membrane is the membrane that actually touches the little hair-like projections on the top, the apical surfaces of our hair cells that are going to cause the deflection of the stereocilia, these hair-like projections, and transduce sound. The other membrane that we can see here that forms another boundary of our scala media is the vestibular membrane, which separates it from the scala vestibuli. And then finally here, the stria vascularis. This is a group of cells that helps maintain the high potassium concentration of the scala media. Because without that high potassium concentration, normal hearing can occur, and sound transduction can occur. What we'll visit in a little bit are the spiral ganglion fibers. So these are the dendrites, the peripheral processes of the bipolar cells that synapse on our inner and outer hair cells. So this is, these are those fibers that are ultimately going to have connections to our first order neurons in the auditory system, which are our spiral ganglion cells. And we'll talk about those in just a little bit. All right, so focusing even more closely on those inner and outer hair cells, we can more appreciate their orientation in this image. Inner hair cells are flask-shaped. They're a little bit more rotund. They kind of remind me of the Florence flask. And our outer hair cells are more cylindrical in shape. Now, looking at the apical surfaces of these structures, in these scanning electron micrographic images, we can see that the stereocilia on our inner hair cells have more of a linear configuration, whereas the stereocilia on the outer hair cells have more of a V-shaped configuration. And how does the transduction of sound occur? Well, we remember the tectorial membrane. So when the tectorial membrane comes into contact with these stereocilia and deflects them toward the tallest of the stereocilia, depolarization occurs. And with depolarization, we have excitation and increased impulse frequency. Now, when they are deflected in the opposite direction, we inhibit nerve impulses and essentially reset our hair cells back to their spontaneously active state so that they are back primed and ready for further depolarization and nerve impulse and sound transduction. But remember, you still have to have a high concentration of potassium for the sound transduction to occur. All right, now let's go back and visit our basilar membrane. So remember, our hair cells are actually resting on this membrane. And imagine unfurling the cochlea, okay, like we're pulling it open. And the basilar membrane is now laid out for us. But remember, this is a coiled structure. But we're unfurling the cochlea, looking at the basilar membrane. So from the base or the top of the cochlea, it is thicker, stiffer, and more narrow. And as you move down toward the apex, it becomes thinner, more flaccid, and more broad. And frequencies are parsed along the length of this basilar membrane, high to low, from base to apex. So think about it in this way. The base is more superior up top, so it does higher frequencies. Apex is lower, more inferior, so it does lower frequencies. It makes more sense that way. But if you've ever blown into a kazoo, you know, like those little flutes, it has that little plastic piece that's really stiff, and it makes a high frequency sound when you blow into it. Same concept here. At the base, your basilar membrane is thick, it's stiff, and therefore it encodes those higher frequencies of sound, as opposed to toward the apex, where it's going to encode those lower frequency sounds. Now, this is a crazy complicated diagram. And I know what you're thinking. I know what you're thinking. Way too fatiguing. Way too fatiguing. So do not learn this diagram, but what I want you to pay attention to is that tonotopy, or the encoding of low and high frequencies, goes all the way up the neural axis, starting at the basilar membrane. And it's lateralized. So rule of thumb is that frequency is typically low to high, lateral to medial. Lateral is low frequencies, medial is higher frequencies. And that's the general rule of thumb all the way to the primary auditory cortex. Same concept. Low frequencies lateral, higher frequencies more medial. All right. So, well, we're running out of time. So I am going to skip these questions. You will have a chance to look at these questions though in Blackboard. So these are just kind of practice questions to look at and that you can refer to on your own time. So real quickly, let's see if we can cover the key players in our auditory pathways. So this is a little mnemonic. Everyone has secret cues superior to learning information. Mnemonics plus acronyms. I know it's a little cumbersome, but it works. So we've already talked about the ear structures. We've already talked about hair cells. We mentioned the spiral ganglia very briefly. So now we've got a whole list of structures to go through. Those include our cochlear and olivary nuclei, the lateral lemniscus, inferior colliculi, and then our medial geniculate nuclei, and then finally our primary and association auditory cortices. So you've already seen this. This is a review slide showing you where we've come from so far. So now let's focus on the organ of Corti in this little box where we see our inner and outer hair cells. We see these peripheral processes coming from our hair cells, giving rise to what's called our radial bundle fibers. So when sound transduction occurs, these hair cells release the neurotransmitter called glutamate. And glutamate stimulates the bipolar primary auditory neurons of these spiral ganglion cells, okay, via these radial bundle fibers. So the signal then is transmitted from these first order neurons in the spiral ganglion cells down these central processes or these central dendrites of these bipolar neurons, which become the cochlear nerve. And then those axons of the cochlear nerve, or that comprise the cochlear nerve, will then synapse on the second order neurons in the cochlear nuclei. So you have dorsal and ventral cochlear nuclei. Now, those cochlear nuclei are in the rostral medulla. So the upper part of the medulla and the lower part of the brainstem. Any damage to the cochlear nuclei and more distal, so cochlear nuclei, cochlear nerve, spiral ganglion cells, inner outer hair cells, any damage to these structures will result in more catastrophic deficits to hearing. So unilateral hearing loss will occur if you damage any of these structures on that side. Anything above the cochlear nuclei, any damage above that will not result in catastrophic losses to hearing or catastrophic hearing deficits, because axon fibers that project from these second order neurons in the cochlear nuclei project bilaterally. They go to both sides, the right and the left, from both sides. So you got crisscrossing of fibers everywhere. And that crisscrossing of fibers allows for communication from left to right, and it crosses in the pons, the caudal pons, in an area known as the trapezoid body. Because the crisscrossing of all those fibers makes a shape that looks like a trapezoid. Now, that bilateral projection then allows bilateral signaling to be transferred all the way up to the primary auditory cortex. So how does it all get there? Well, after some synapses in the superior olivary nuclei in the caudal pons, you then have transmission of impulses through fibers known as the lateral lemniscus. So some of you have heard of the medial lemniscus, I think. I think y'all have talked about that. Lateral lemniscus is with hearing. So now these fibers are going to carry signals up to the inferior colliculus in the caudal midbrain. And there will be synapses there as well. So from the inferior colliculus, fibers will then travel through the brachium of the inferior colliculus to another nucleus in the rostral midbrain. This is known as the medial geniculate nucleus. And then from there, fibers will run through what's called the auditory radiations inside the internal capsule, and then reach the transverse temporal gyrus, which is the anatomical name for our primary auditory cortex. Okay, so lots of synapses, lots of structures, and really the order of neuron can only be named if a synapse occurs between, the axon and the cell bodies in that nucleus. It's very complex. This is really an oversimplified explanation of it because you've got projections going everywhere and connecting with everything. But if you go back to that mnemonic, that's really kind of the general simplified sequence of structures in the auditory pathways. And yeah, I've kind of already talked about these. So that's not the end of the story though. You also have association cortices, Broca area and Wernicke area. We take off the possession, the apostrophe S, because, those scientists that discovered this, they don't really own those areas. So we just take off the apostrophe S. Broca area is used for speech production. Wernicke area is used for speech comprehension, which is why hearing is so important to producing and understanding language. This is why people who have hearing deficits and it's not recognized, they will oftentimes have speech deficits that will follow them later in life if those are not corrected. And so all of these are connected through fibers known as the arcuate fasciculus. And here I've overlain on the side here some brainstem slices that have been stained. So you can see what these slices would actually look like, from an anatomical perspective. All right, I'm going to stop there because we have gone over time, and I'm just going to let y'all answer some questions. And we'll just not worry about the rest of the content in this lecture. Any questions? Since there just was not enough time to get through the other content areas, I'll just say you're just not responsible for knowing that. One of the concepts that we didn't get to is sound source localization. So where is a sound coming from in space? That's why you have two ears. So just, for the sake of understanding, if a sound is coming from this side, I know it's coming from this side because my other ear tells me that it's taking longer for the sound to get here. So you need that cross communication between both ears in order to figure out where is the rabid tiger coming from in the jungle that's chasing me, so I can, know when to run and really when to run and get away from it. And all of that is really the superior olivary nucleus is in control of that sound source localization. Any questions?
What are you thinking the format of your questions will be?
So if you look at the example questions, and I don't know, Dr. Syed can inform me if I'm in fact writing my own questions. I don't know if I am or not, but if I am, I would follow the format of the examples I have in this lecture and then in the slides that are currently in Blackboard.
So let me make it clear, Dr. Meyer, you will be writing question for 2 of your lectures actually.
Okay.
So that's your part.
Okay, so what I'll do is I'll follow more of a format for those example questions that you can see in the slides.